In this video, we are going to deal with how to evaluate scatter plots. Scatter plots are used to show the relationship between two variables, sometimes with mathematical modeling. Individual dots, representing samples or averages, are plotted on a graph, and then statistical software analyzes whether there is any sort of mathematical relationship that can be used to describe the data. Scatter plots can be generated with both individual data points or as averages. Just remember, if the spots represent averages, you should always have error bars. Once the dots have been plotted, a line of best fit can be drawn. This is what describes the relationship between the two variables. An equation that describes the line of best fit can be generated, or you can simply describe the relationship. In this case, we would describe this as a linear relationship, since the line of best fit is a true straight line, and we would say there is a positive correlation. That is, when one variable increases, so does the other. In this case, we are seeing the opposite, a linear relationship with a negative correlation. As one variable increases, the other variable decreases. Also, the relationship does not always have to be linear. This is a graph showing an exponential relationship between time in hours and counts of bacteria. This is still a positive correlation as both variables increase together. One thing to check for with a scatter plot is whether or not they have enough data to cover the range of their line. This graph to the left has a line of best fit where there is data covering pretty much the whole length of the line. This is what you should look for. This graph on the right is a bad example. All of the data is clustered to one end with a single outlier way over here. We can't be sure that the same linear relationship holds true in the middle region, as we have no data to support that claim. Also, with any line of best fit, you should be looking for something called a correlation coefficient, also reported as an R-squared value. This is a measurement of how well the data actually fits the line. A perfect correlation has a value of 1.0. This is the highest value possible for R-squared. The lower the R-squared, the poorer the fit is between the line and the data. When someone is claiming a specific type of relationship, they should report their R-squared values so you can interpret how good their data actually fits the relationship they are proposing. To the right are 3 lines of best fit for 3 specific genes, looking at the expression of those genes and a gene called HSP70. All 3 have high R values, suggesting the linear relationship is a good description of the data. Also, data points are available for each gene across the tested region. Finally, I will point out now, and again later on this semester, that correlation does not equal causation. Just because 2 variables have a mathematical relationship doesn't mean that one depends on the other. In this case, the expression level of HSP70 might predict the expression level of any of the other 3 genes, but that doesn't mean it actually causes that gene to be expressed at that level. And now for a bad example. This paper comes to us from the website Mercola, a website known for publishing pseudoscience. This link is reporting on a study that looked at maternal exposure to fluoride and IQ in children. Fluoride is added to drinking water as a major public health initiative to reduce cavities. Its safety has been demonstrated for decades. This study tries to suggest that is not true, and that when moms drink fluoridated water, it could lower the IQ of their children. So let's look at the actual study this post is based on, and the graph that appears to show a negative correlation between maternal exposure to fluoride and child IQ. This is a scatter plot with a line of best fit. But where is the correlation coefficient so we can see how good the data fits the line? It turns out the authors of this paper don't provide one. However, we don't have to take their claims at face value, as there is software out there that can take a graph, pull the data points from it, and allow you to regenerate published graphs and the underlying data for them. This is what you see here. The graph on the right is one that I generated, and you can see how it matches up with the published graph. So now that I have the data pulled out, I have the graph, we can run statistical analysis and actually calculate an R-squared value. So what is the correlation coefficient for this data? The R-squared value is 0.0301. This is an incredibly low R-squared value. And it means there is no real correlation between these two variables. This makes sense, as if I look down here at this particular exposure to fluoride, I can find individuals with very low IQ, individuals with high IQ, and all sorts of values in between. So the exposure to fluoride is in no way predictive of childhood IQ, which is exactly what this paper is claiming to be the case. To summarize, when you want to critically examine data, there are several questions you should ask yourself. First, are there error bars, or at least a reported standard deviation? If averages or other ways of aggregating data are reported, so should error bars. Second, you should look to see if the x-axis and y-axis are appropriate and scaled correctly. Third, look at the p-values reported for that figure and determine if there is any real difference. Fourth, look at effect size and determine if the difference reported is meaningful. And finally, for graphs looking at correlations, make sure you look at what the correlation coefficient is and whether or not their data truly is described by that type of relationship.